Thank you for being here for today's webinar talk on the mechanisms of circulating tumor cluster metastasis by Dr. Tyler Allen. Dr. Allen completed his Ph.D. studies in comparative biomedical sciences at the North Carolina State University College of Veterinary Medicine and is now a postdoctoral science at Duke University Cancer Institute. Dr. Allen has been featured in the Forbes 30 Under 30 list in the science category last year. Dr. Allen is also a winner of the GENEWIZ Traveling Wizard Grant for 2018, where his project focused on using ultra-low RNA sequencing to identify key mediators in circulating tumor clusters leading to metastasis, which was recently published in the Journal of Cell Science. Please help me in welcoming Dr. Tyler Allen.
Thank you, Jesse, and thank you all for having me here, and thank you all those who are joining us digitally. So today I'm going to be talking about the project that we recently completed in conjunction with GENEWIZ here, and it's titled Utilizing Ultra-Low Input RNA Sequencing to Investigate Tumor Metastasis. The goal of this project was to just look at tumor cells that were migrating or metastasizing and then see or find out unique genetic profiles about what made them different and what allows them to form new tumors at different sites. So I'm currently a postdoc at Duke, but like Jesse had mentioned, this work that I'm going to present on was from my dissertation at NC State University's College of Veterinary Medicine. So real quick, so just the background of what the study was. So I kind of mentioned it. So this is a nice little outline that I like to give for the study. And it's pretty much showing what the major findings were of the study. And it's that tumor cells that travel and metastasize or exit the circulation can do so in a few different ways, but the ones that exit as clusters versus as single cells, the clusters seem to be more likely or more able to form secondary tumors or new tumors. So these are the ones that are most dangerous and that have the most kind of potential for looking at in terms of developing cancer therapies or anti-metastatic drugs. So what we wanted to do specifically was look at we want to isolate or capture these cells that are growing versus the ones that aren't, and then see at the genetic level through sequencing how they differ at the mRNA expression level, and then what's causing them to be able to both exit and then form the new tumors. But we were also interested in looking at the blood vessel cells as well. So the blood vessel is comprised of what's known as endothelial cells, and so these cells actually contribute to the process when cells are exiting the circulation to spread to new parts. So you want to look at kind of a dual system where we look at both the tumor cells and see the genetic components that are different in them, and then also the endothelial or blood vessel cells that are contributing to this process, and then what's happening at the genetic level to allow this or incorporate this. And then this is where GENEWIZ comes in to save the day, because we worked with GENEWIZ or with you all to utilize you all's ultra-low input RNA sequencing, which allowed this study to be possible. So I'll get into a little bit background. So if you're not familiar with metastasis, that's just when cancer spreads throughout the body, and it's pretty much, it's kind of the most dangerous part of cancer. So most people who have or get cancer, they won't really suffer or die from the cancer unless it spreads. So when a person has an isolated single tumor in a single area of the body, that's bad, but once it spreads, that's when it gets really bad and very dangerous. And it does this by having like a primary tumor. So say you have a tumor, let's say in your lungs. Tumor cells or cancer cells from that tumor will break off from the primary tumor into your circulation. They'll travel through the bloodstream, and then they'll exit at what's known as a secondary site, like say from the lung to the kidneys or the liver, and then they'll begin to grow, or sometimes they'll begin to grow and form what's known as secondary tumors, and then that's when usually cancer patients see a lot of difficulties or troubles when the cancer spreads to multiple parts of the body. So I say that to kind of underline or emphasize the importance of understanding this process in cancer research, the metastasis process, because that's where most of the problems occur. So the big thing one of the big things that we found with this study, so this is just a diagram that shows we found that cancer cells are actually pretty tricky. They can exit or travel through the circulation and exit blood vessels in multiple ways. There's a way known as Angiopellosis that we discovered where the blood vessel will actually wrap around and then push the cell out, but also cancer cells can kind of squeeze through the blood vessel and then exit. And so we found that, or it's been shown that cancer cells are actually kind of smart in the sense that they can find multiple different ways to achieve what they're trying to do, which makes them so dangerous. But the important part, so to investigate this or to study this in a model, we use zebrafish as a model. And so specifically we use zebrafish at the embryonic stage. So these are just a few days old. And the reason we use zebrafish is because, one, they're at the embryonic stage, they're completely transparent. And then we have a genetically modified strain where all the blood vessels inside of the zebrafish are fluorescent with either a green fluorescence or a red fluorescence or some other type of fluorescence. So what you're able to do is inject tumor cells directly into the zebrafish, and then in real time, you're actually able to see or modify or track the tumor cells travel through the circulation of the zebrafish, and then most importantly, you're able to see how and when they exit the circulation and then exit the blood vessel and then start to form new tumors. So we're able to model this process in zebrafish so that we can better understand how this is occurring or how this is happening in humans, and so we can better develop therapies or treatments to target or to prevent this from happening. So what we're able to do is once we inject them into the zebrafish, we take it over to what's known as a light sheet microscope, which is just a fancy microscope that's able to take a thin kind of sheet of light and I scan the zebrafish while it's still alive over the course of several hours. So what you're able to do is take zebrafish that have tumor cells injected into them and then image them for hours, sometimes even days, while the zebrafish is still alive. So you're able to see in real time how the tumor cell is interacting with the circulation and the zebrafish cells and the blood vessels all in real time. It's able to give us some really cool images and really cool videos I'm going to share with you in just a second. So the next thing I'm going to show you is a video of what I was describing. So I showed you the animation, and so this is actually a real-time video of what I was explaining in the zebrafish. So the red that you're seeing here are the actual blood vessels, and then the blue or these cyan-colored dots are the tumor cells or cancer cells that have been human tumor cancer cells that have been injected into the zebrafish bloodstream. And so this one, we're going to see the video of the tumor cell undergoing angiopellosis, where the blood vessel is kind of coddling and pushing it out. And then this one is an animation of, again, the other method where it's squeezing through a process known as diapedesis. And then on the, I guess, the right side, these are 3D reconstructions of both of the same videos. And so this is important because, like I said, we've shown that, or we've seen that tumor cells can use multiple ways to exit, but this is where it gets really interesting, because what we found later, what we found next, is that tumor cells that use this method, the angiopellosis method, to exit can actually do so, one, as single cells, where it's just one cell exiting by itself, but they can also use this method to exit as groups or as clusters, which is just when you have two or more tumor cells that are kind of traveling as a pack or a group and they leave together. So here's another video that shows that. So here, this is a group or pretty much a glob of what's known as circulating tumor cell clusters. And it's going to see over the course of several hours, it's going to go through the angiopellosis method, and there's an extravascular cavity that's going to form here that they're going to be pushed out into. And if you think about it, this is what's happening in humans when they have tumor cells that are traveling. They have the ability to travel as clusters, and then once they exit like this is when it gets dangerous, because this is when they form new tumors or secondary tumors. So there's a lot of merit in studying this process, because it will allow us to better understand what's going on and how we can prevent it in humans. But so we didn't want to, so this alone was really interesting to us, just the ability or the fact that we had discovered this new method or mechanism that tumor cells could use to exit the blood vessel, both as single cells and as clusters. But we wanted to take it a step further and then look at specifically what about these tumor cells, these clusters, was causing them to have an increased ability to form secondary tumors or form new tumors in the body. So what we did, here's actually, before I get into that part, is just another video animation that shows the effect. So this is the cluster cell versus the single cell that kind of travels through. And what we saw in the zebrafish model is that they both exit, and they both exit together, or they're both able to exit through the similar manner. But what we're going to talk about in a second, one of the most interesting parts of this was that we found, or we saw, that tumor cells that exit as clusters show an increased propensity to form new tumors versus a cell that just exits by itself. Kind of like, I use the analogy, when you're in class or when you're in high school or college, when you're with your group of friends versus when you're alone. You might act a little bit more rambunctious or unwildly when you're in a group or a cluster versus when you're by yourself. So it's kind of, I use the analogy with cancer or tumor cells where it seems that they exhibit a similar behavior where they're much more likely to be dangerous when they're in groups or clusters versus when they're by themselves. But interestingly, the clusters only seem to be able to exit through the Angiopellosis method. So it's the other method what I showed where the diapedesis where we're squeezing through. As you can imagine, it'd be kind of hard for a big, huge cluster of cells to squeeze through. So that method is not possible for the clusters to exit through, only through Angiopellosis. And I'll tell you in just a moment why that fact is important. So what we did to study, or how we studied this, the growth rates over time, is we injected cells, the tumor cells again, to the zebrafish, and instead of looking at only when they exited the blood vessel, we took it a step further and we looked at what was happening after they exited, and we watched them or we observed them for several days after they exited. And so here you see here's an example of just one single tumor cell that had exited, and then again the same tumor cell after 4 days. And so you can see it's still just one cell. It hasn't grown. It's kind of remained just dormant or stagnant in its growth or its proliferation. And this is interesting because this type of tumor cell, this type of cancer cell, usually grows or divides rapidly, like very quickly. So it's a bit unusual, or it seems like it would be unusual that this cell wouldn't have grown or wouldn't be at least multiple cells at this point after 4 days. And so we did a similar experiment where we injected groups or clusters of cells. So here is a cluster of 3 cells that are injected, and they're already extravasated, and they're in an extravascular cavity right now. And then again, so we looked at 1 day versus 4 days, and you can see over 4 days, there's actually a huge or extremely significant increase in the growth rate or the amount of proliferation in the size of the tumor cell cluster. And we saw this across the board with a lot of different cancer cell lines in both human, we use mice as well as zebrafish and other types of cancer cell lines. And we saw a similar trend where tumor cells that exited alone always tended to remain single cells or remain dormant, is what it's kind of known as in the clinical field, whereas the tumor cells were much more likely to form new tumors or to grow if they were exiting as clusters. So then we wanted to take it one step further and see, I guess, not only what was happening after they exited, but we wanted to isolate or specifically take some of the cells that had exited and then see at the genetic level what was happening or why were these cells growing or what potentially might be causing them to behave differently versus these single cells that don't seem to grow or cells that just never exit the blood vessel at all. What was the genetic difference? And then, so what we did, so this is where we kind of, at first had ran into a roadblock because, so I'll go back to, so this one, so what the plan was when I had originally thought of this, so I was like, okay, the cells are fluorescent. So what we do, we just inject a bunch of tumor cells into the zebrafish that are fluorescent, and we observe which ones that exit. So what I wanted to do was use some sort of fluorescence activated cell sorting, or FACS, to get them and then sequence them. But the only problem is, if I were to do that with either of these types of systems, there are going to be other cells that are fluorescing that either that haven't exited, or that maybe aren't growing, or that aren't the right type of cell that I'm interested in. But the thing about the zebrafish embryos is that although it's very blown up here, the embryo is about the size of a period. So when I'm doing these injections, I'm using a micro injector, and it's a very fine, like almost they're literally the size of a period, and you use an injector and you inject the tumor cells directly into them. So it's not like you're able to dissect the tumor cells out that you want and only use those. But if you use FACS sorting, then you'd get all of the cells that are within the zebrafish, which would be a large population that aren't the ones that I want. So what we did is we we developed a new system or new model where we use what's known as a photoactivatable or photoconvertible fluorescence protein. And that's simply just a protein so you can transfect it or transduce it into a cell. And it's for this one, it originally is a GFP or green fluorescence. And then with targeted UV photoactivation or activation, you're able to irreversibly change that fluorescence from GFP to RFP. So what this allowed us to do is take a targeted piece of, so say we have, go back to the other slide. So we have these tumor cells that we're interested in getting, but we don't want some that are around here. What we're able to do is we could repeat this experiment with tumor cells that were fluorescent with the photoactivatable fluorescence protein, do a targeted UV stimulation of just these tumor cells, and then once these have been changed from a specific fluorescence to another, then do FACS sorting or the fluorescence activated cell sorting to get only the specific population that we're interested in. But the problem with that is that, so here it's just a few cells, like very few, and even if you pool a bunch of experiments together, you're still only going to get like a few hundred, maybe 1,000 or so cells, which is typically much lower amount, or far too low of amount to get the type of sequencing, RNA sequencing that's done normally, which is where we, GENEWIZ came to the rescue because you all have the ultra low RNA input sequencing. So I talked with the team here, and we had kind of discussed, when I was telling them the project that I was doing, they'd work with me. And it's funny, I was speaking earlier, so the project, or the way that I was thinking about doing it, was actually different from how we ended up doing it, because it changed when I talked to the team here at GENEWIZ, because they helped me to kind of cultivate the way I was going about this much better to allow us to see the type of sequencing data that we wanted. So, but essentially what we did is we took the, so we had two different models. So we wanted to look at, again, both the tumor cells that are exiting, and then, like I mentioned earlier, the endothelial or the blood vessels. So we've got two different systems. One system was the zebrafish system, and we had a model or a different line that was transduced or had only the photoactivatable fluorescence in the blood vessels. So what we're able to do is, again, just kind of look at through the microscope, look at the cells as they were exiting the blood vessel, and then immediately after they had exited the blood vessel, we're able to do the targeted UV photoactivation on just the endothelial cells or just the blood vessel cells that were participating in the tumor cells extracting or extravasating or exiting, and then isolate those with and submit them for ultra-low RNA sequencing here. And then again, kind of a similar thing, except this time, instead of the endothelial cells, we focus on the actual tumor cells. So the tumor cell clusters that exited as groups right after or immediately following their exiting, we use targeted photoactivation and then change the fluorescence, and then use FACS sorting to get the cell populations that we were interested in, and then we use those for the sequencing here at GENEWIZ. And then we got lots of really cool data I'm going to show. So the team here at GENEWIZ did a lot of the analysis as well, and so I'm not going to go too detailed. The analysis that was provided here was very detailed and much more extensive than what I'm going to get into, but I'm kind of just going to show you the overview, or kind of the details of, or not the specific details. But so we looked at differential gene expression between the two populations, both of the endothelial cell population that was isolated for the tumor participating endothelial cells, as well as the tumor extravasating cells that were the ones that were exiting. And we were able to find a myriad of different genes that were differentially expressed in both of the populations, which has never been seen before, because this type of study is kind of the first of its kind, where we're looking at, so a lot of times in the clinic or in hospitals, if a patient has cancer and it's spread to different parts of the body, doctors or scientists will take what's known as a biopsy or a piece of that tissue, and they'll study that. But the thing about that is, by the time a secondary or even a primary tumor is detectable, it's already grown for probably a long time. So it's almost sometimes a different type of tumor expressing different genetic profile from when it first exited or from when it first started traveling throughout the body. So this method allowed us to see in real time, and specifically the key point when the tumor cell is exiting the blood vessel to when it's about to just grow and start the initial genesis of the new tumor, what the genetic profile of that is, because that's the key step to understanding metastasis, is when it's about to exit and then form those new tumors. What is the genetic profile then? That's what we're interested in. And that's where I feel like is a lot of potential in terms of the cancer biology field and therapies for metastasis, is understanding that process. So with the help of GENEWIZ, we were able to identify a myriad of differentially expressed genes that we think are excellent candidates to study further in terms of what their actual functional role is. And that's the kind of the next steps of this study after the, I guess, what we've gotten. So what, I guess the kind of takeaways that we got from this study is that the tumor cells which survive the whole metastatic process where they exit and they travel to a different part of the body, they have, or they exhibit specifically unique genetic profiles, and specifically cells that are traveling and exit blood vessels right when they exit or during the extravasation process. They're exhibiting unique profiles that are most likely contributing to their ability to, one, exit blood vessels, and then two, form new tumors at distant sites. And then also, it's not just the tumor cells itself that are involved in this process, it's the endothelial cells of the blood vessel that's also contributing to this process. So and this is really the, I guess, the first type of study that was able to isolate endothelial cells that were participating in this, because one of the limitations of imaging technology now is that we don't have the technology to do the type of resolution of live imaging in humans that we need to be able to see tumor cells traveling in the circulation. So there's no way to get or to receive or to isolate endothelial cells that are participating in the method, because there's really no way to see them at the level that you need to. And then even if you could see them, get them from the actual patient who has a tumor that's involved in the process. But so from here, the future studies will involve using the gene set, or the gene set and analysis that was done here at GENEWIZ, to look further at the functional role of these specific genes, and then see how we can either modulate or leverage this information to develop new cancer therapies, or just better understand the metastasis process, which will hopefully in the future allow us to understand what's going on and prevent this from happening in cancer patients or people who have maybe a primary tumor that hasn't spread, or if it has spread, understand how we can prevent or decrease the amount of secondary tumors that form, so we can hopefully save lives and help people who have cancer or who may get cancer in the future. And then, so if you are interested in learning more about any of this study, it was recently published in the Journal of Cell Science, so feel free to check that out. And then lastly, I want to give an acknowledgement to all of my lab members at my graduate institution, as well as our collaborators, and of course the great folks at GENEWIZ here who were really instrumental in this project being completed, both on the analysis side of things, the performing of the sequencing, as well as the planning of the steps or the project to get the type of data that we needed to help advance this, the understanding of this process. And also the NIH and the National Cancer Institute and the National Heart, Lung, and Blood Institute who helped fund this research as well. So thank you so much, and I'll be free to take any questions.
Thank you very much for the talk. It's really interesting. I'm just wondering, could you share more maybe one of the top upregulated or downregulated genes that you find very interesting and maybe you're considering looking into it further?
So there are literally thousands of genes that were showing differentially expressed levels. And one thing I didn't show, part of the analysis that GENEWIZ did, is they actually looked at the did gene ontology analysis. And so it was looking at pathways that were differentially regulated. And one of the interesting pathways, or one of the few interesting pathways, or many interesting pathways, was there was cell adhesion and ion transport pathways that seemed to be differentially modulated. So we were looking more, that's of course interesting because one, with the cell adhesion, of course for both cell types, the clusters, they're adherent to one another. And then also the endothelial cells, that process is pretty much cell adhesion, and then there's a lot of ion transport in that process of the modulation of endothelial cells. So I think that those those types or those pathways, not specifically genes or say, but the genes within those pathways and the pathways or the genes that show differential expression in those pathways are the ones that would be good to further pursue and then look at how those are behaving, because I feel like there's a lot of potential in terms of, we already know that cell adhesion and ion transport and P10 signaling, all things that were modulated, have shown correlation to cancer and cancer progression. So I think the next thing is to look at it in this system or in this model and see how, if we either knock down or modulate certain gene expression, it'll change the phenotype of what we're seeing.
So I wanted to know, what is the percentage profile difference between the primary tumors and the ones that you found exiting from the blood vessels and going on to make other tumors? So what is the difference in their genetic profiles?
So it's the primary ones that we saw. It was a lot of changes again in like cell adhesion. There were two major pathways that seemed to be differently expressed. It was the P10 signaling as well as some of the other, cell adhesion and then modulation of ion transport. So those seem to be the biggest ones, but we're still, the thing about this type of data is it's kind of a vast amount, so we're still, deciphering which ones are the best kind of key things that are different between the two populations. So right now we kind of have a general sense, and that's kind of the basis or the seminal part of the study. And then the next parts will be kind of diving further deep into seeing what distinguishing factors or what's causing or what the effect of these distinguishing factors. So it's a bunch or it's a lot, but it's all in the supplemental figures in the paper, but it's far too much to kind of distill into a short short talk. But it's a lot of interesting stuff, though. I'll say that it's a lot of interesting areas.
Great talk. I was interested in what causes tumors to exit at specific sites. Is there something within the endothelial cells that causes them to exit there, or is it just kind of random?
That's something that is still a bit contentious even in the cancer biology field. So there's thought, or some of the thoughts are that there's some type of signaling that's occurring either at a chemotaxic level where the cancer cells are able to either detect or sense some type of chemical or some type of gradient, and they're able to, similar to certain stem cells, travel to or detect a specific spot and then only go to that area. But so like one thing with cancer cells, certain types of cancer cells, if they metastasize at all, will metastasize to completely different spots. So if you have a breast cancer versus lung cancer, both of those can metastasize, but usually the spot where a secondary tumor will form for a lung cancer will be different from breast cancer. So it's still a lot of, information or studies that need to be done to really pinpoint why tumor cells are exiting or metastasizing to different spots. But a few of the theories are that there may be tumor cells that are exiting at multiple different areas. It's just they're not able to survive, or they're only able to grow and form secondary tumors at specific spots. So we see the tumor cells that grow at those sites or those specific sites, but there may be other areas where they are extravasating or exiting the blood vessel, but they're just not detectable, or we don't ever find them.
When you were studying the zebrafish, were there specific spots where the injected tumor cells tend to prefer to exit as opposed to other areas where there was less often exodus?
So that's an excellent question. So one of the limitations of using a zebrafish model is that, so it's not as, so the vasculature and the circulation of the circulatory system of the zebrafish is much more limited and kind of a basic level compared to humans or other animals. So it's kind of, when you inject them into the, you inject them pretty much right before they enter the heart of the zebrafish, and then they'll travel through the heart, and then kind of they'll just go in the circle and like kind of just circle around, which is much more basic than the complexities of the human circulation. But for most of the imaging or most of the extravasation, we identified that it happened in pretty much the tail region, or these intersegmental vessels of the tail region, and then kind of the larger portions. But we one of the limitations also of the imaging system is that the further you go up, the more opaque the zebrafish becomes, so it makes it much harder to do the type of high-resolution imaging that we were doing. So most So I guess the caveat is that most of our imaging was done down here, which is subsequently, I guess, where we saw most of the extravasation. But we did see, or there have been instances where we've seen extravasation occurring at different areas. So I don't think that there's, at least for this cell type, there didn't seem to be a specific location dependent area where they extravasated. But I think that that might just be a result of the limited exposure or the limited ability for them to travel to different complex places just because the system is a more basic system. So I feel like that's an interesting question, but we're more so just interested in the extravasation or the exiting process, not necessarily the location, which I think is also an extremely important question, but might need a different system to study that part better.
So you talked about the two methods for exiting of the tumor cells. Is one method preferred over the other, and if so, why? And if not, so why?
So yes, that's an excellent question. The two methods, there's the Angiopellosis where the cells are actually being coddled and kind of pushed out, and then the other, the diapedesis, like where they're squeezing through. And to answer your question, so we saw that the one where they were being coddled and pushed out, the Angiopellosis method, that one showed a markedly increased ability, or the tumor cells showed an increased propensity to exit through that method versus the squeezing or diapedesis method. And then secondly, the tumor cell clusters were exclusively only able to exit through the Angiopellosis method and not the diapedesis method at all, at least from our observations. But to answer the second part of that, why that's occurring, that's another good question that we're still working out. So it's a few hypotheses is that it might be less taxing for either the tumor cells or the endothelial cells to go through that method versus the diapedesis method, even though it seems kind of counterintuitive, because when you have the vascular restructuring, you would think that that would be more taxing since it's actually a restructuring of the vasculature in the endothelial cells versus a cell just squeezing through. But it seems like at least tumor cells prefer that method, potentially because they're able to exit as clusters and then form secondary tumors higher propensity of secondary tumors. But it could just be that there's some type of evolutionarily conserved method that method for some reason was the primary method that tumor cells, or even just cells in general, because other cells have been shown to use this method as well, used to exit when they're in the circulation that aren't native to the bloodstream.
I had a quick question. First of all, really great talk and really great research. My question was, you talked about photoactivated cells, right? Can you talk about how you made sure that the photoactivated cells were like for the clusters rather than the single cells? Because I think the target was to look at the clusters, right? Mm-hmm. Can you talk more about how you made sure or ensured that clusters were the ones that were photoactivated?
Yeah.
Thank you.
So that's a great question, and it was something that was actually really difficult. So what we did is, so we had two different systems, and I might go back and show this. So that was something that we were really focused on because it was of extreme importance to only get the clusters and not the ones that we were not interested in. So what we did is, so we used a microscope, and then pretty much with the photoactivation under the DAPI wavelength is what you use to activate or do the conversion from one fluorescence to the other. So what we had to do was to define, so what we did is we visually would look and see the cells or the location of the cells and say, for instance, some of them, like this is a good example because it's kind of just the cluster by itself, but some of them it would literally be, there would be a cluster here and then like another single cell like right here. So if we photoactivated the whole area, then that tumor cell would also be activated. So what we had to do is play or kind of adjust, find the aperture and some of the settings on the microscope to hone in and specifically capture or decrease the amount of UV or specify the UV activation to specific area or length, and then only allow it to activate. So instead of it being just in a complete activation, we would hone it in and then completely only activate the cells of interest without or with removing or making sure that we weren't photoactivating other cells that were in the vicinity that weren't either extravasated or the ones that we wanted. So it was a very tedious process in some sense because we were doing this with, again, hundreds of zebrafish, so we had to go and manually look through, I had to go and manually look through all these zebrafish and look and find the ones, and then this was I guess, super high magnification and then activate only the ones that were of interest and then go and then do that subsequently for hundreds and hundreds of fish. But that's how we achieved that.
So thank you for sharing your research, Tyler. So I remember you said you noticed there were changes in endothelial cells after interacting with the tumor cells, right? Did you go into deeper details of what changes? Because I am wondering if these changes make those sites, those particular sites, more susceptible for the tumor cells to act as exiting sites.
So that's an excellent question. And I think for me, when I think about it, I think that or at least how we were kind of positioning after getting the results is we were thinking that the endothelial cells were not necessarily, they weren't changed to predisposition them to allow the tumor cells to exit, but I was thinking more so that after they were interacting with the tumor cells and exiting, or during that process, there was some type of change that was occurring or that had occurred during the process. But it could be true. It might be that what we're seeing is that those endothelial cells already had a genetic predisposition or a genetic change or profile that allowed the tumor cell to then interact in a certain way and exit at those points. But that's actually a good perspective that I hadn't really thought about too much before. But I think that with this type, or I guess with the conclusion of this study, that'd be something good to focus on next in terms of, so we do have the sequence or the results of the differentially expressed genes, and then seeing or modulating those in maybe a zebrafish model or some other model with in vitro endothelial cells, and then seeing how that affects the ability, one, of cancer cells to interact with those, or two, looking at after they interact with those, is that when the genetic changes are happening? Because we did see, those were some where I mentioned with the ion transport and cell adhesion, which is I guess a known process in the metastasis process, or allowing cells to exit or travel through the blood vessels. So it could be what you're saying is maybe why they're traveling at those specific sites is those endothelial cells could be genetically predispositioned to allow that. That's true. We need to collaborate. That's an excellent idea. Yes, that's a possibility. But I think the way I was initially thinking about it was that the endothelial cells were changing as a result of the interaction with the tumor cells. But that could be a possibility. So once I figure that out, I'll let you know. Thank you.
So I'm curious why the clustering is so important. Do you think it's really necessary to have clusters to exit the blood cells, or is the clustering just a symptom of a genetic profile or transcriptomic profile that's really important for exiting the blood vessels?
So I think that we see that both the single cells and the clusters can exit just fine. They're both able to do it, and they do it very frequently. But the thing about the metastasis process is that it's a very inefficient process. So if you think about it, you've got a primary cell that's native to a specific organ or specific location in the body. Then it's pretty much jumping into the circulation, which is analogous to us jumping into a raging monsoon of water. And that's extremely rough on the cells. So then they have to travel through this monsoon, and then kind of they're exiting at a different location. And then once they've exited, they're at a completely foreign land. They have no ability to know or to modulate where they exit specifically. So they're at a place where they're not biologically meant to be. So it's very hard, or it's very harsh on them to go from a completely new, or from an environment they're familiar with, to a new environment. And so I say that to kind of postulate the idea that the cluster is potentially allowing the cell to bring with it some of the seed and soil theory with cancer metastasis, where it's bringing some of its own soil to this new location, which could potentially be allowing or helping it to become better acclimated to this new environment. Because even although I mentioned metastasis is it's usually where things become most dangerous, it's actually only a very small percentage of cancer cells are actually able to successfully metastasize and form new tumors. It's a very tiny percentage. It's just that those ones that do are usually the most deadly, so those are the ones we focus on the most. But so to answer your question, I think that it's more so, it's not that the clusters necessarily have some type of huge genetic advantage over the single cells. I think that they both utilize a similar type of process and may express similar or varying genetic profiles, but more so that it's the the profile that's allowing them to be happy with their friends at a different location that's giving them the ability to form new tumors more efficiently, because they're both able to exit. It's just one's able to survive better. And I think it's because cells like to be near cells of a similar kind, and they're just happier, so they're more dangerous.
So you mentioned you used several different cancer cell types. Were there some that had a propensity for clustering over others? And did you also look at primary tumors as opposed to cells that have been passed over a long period of time?
Yeah, so excellent question. So we did see differences in the, amount of clustering and extravasation between certain cell types. For this study, we were focusing all on commercial cell lines, or kind of available cell lines that were commercially available. So we did, I think in the future, it definitely would be good to look at primary cells and then look at the difference of the changes in those. It's just the, the access to those types of samples is a bit more tricky for, kind of the regulation and stuff like that. But so I do think that one thing would be interesting to look at is looking at the genetic profiles of different types of cancer cell lines that undergo this process and the ones that do it differently, and then seeing if those genetic expression levels or any of those genes are different that might be correlating to a change in both the clustering of the cells and then their extravasation or exiting potential for the process. But I think that we definitely, at least at the initial stages, see differences in the behavior of cells in this process depending on what type of cell it is, the location and the quantity of the cells, and the terms of how well they cluster both in the system and then during the process. So I think there is definitely some at least phenotypic changes that we can visually observe or see and quantify between cell lines. But I think it'll be even more interesting in the future to look at this at the genetic level and then see what's happening. And that might open up some more insight into what's going on in this process, if we can understand or see a cell that is good at it and then see what that genetic profile is compared to one that also does it but maybe worse or less frequently doing it, and then seeing how that changes the modulation of what we're looking at.
This is a generic question. Given the vast amount of experience you have in studying cancer and the vast amount of research we are having in cancer, what do you think is the rate limiting step? Why haven't we able to make as much progress as we have shown in the process of studying or understanding the mechanism of metastasis or just cancer in general?
That's an excellent question, and that's one I get a lot from especially like people who are certain like family members and friends I have who aren't familiar with it, and even in the field it's a interesting question. But the general question of cancer, I guess why maybe we haven't progressed as far as with cancer is maybe we would want to, is that the way I like to think about it is with cancer, we say cancer, but cancer is almost like an umbrella. The way I feel like it's best described, cancer is an umbrella term for a myriad of multiple different diseases. So even each cancer, even if it's the same type of cancer or the same location, between patients, there can be a complete difference or change in the type or the etiology or the cause or the genetic background of that cancer. So say person A has lung cancer and there's person B, they also have lung cancer. The genetic cause or the genetic components of those cancers will be completely different. So even if we are able to understand something about person A's cancer, it may not hold true for person B's cancer, even though they're the same type of cancer, same location. And I think that's one of the difficulties of cancer research, is kind of the magnitude of differences or change or causes that can be affecting. And it's not just one thing. Usually with cancer, it's an interplay of multiple different things happening simultaneously. And I feel like that's what makes it so hard. But that's also one of the things that makes it so interesting to study, is that there's so many things going on. And if you're able to look at some of those things and how they correlate with one another and then advance it somewhat, that's at least something. But I think to answer the first part of your question, I think metastasis specifically, that's a challenge in and of itself, because it's like I mentioned, it's very hard to see the metastasis process in humans or most animal models. By the time we're looking at tumor cells that have metastasized, they've grown or they've already been at that secondary site for such a long time that they're detectable. But also, with cancer cells, there's a lot of heterogeneity. And by the time, if they're growing and growing, by the time that they've grown to a detectable level, it may be completely different at the genetic level than when it first was pure, or when it first metastasized. So I think that's one of the hardest things. So even if we are looking at tissue or samples that have been from metastatic patients or patients with metastatic biopsies, the genetic profile of what we're seeing might be different from what's actually causing or allowing those cells to metastasize. So it's stuff like that, where it's just there's a multitude of different components and steps that are happening all in tandem that make it such a difficult thing to pinpoint specifically certain things that are causing or affecting things at the biological or genetic level. But I hope at GENEWIZ we're going to figure it out. Don't worry.
So Tyler, I was very curious when you said that you were injecting single cells versus the main aim was to actually take in and inject clusters. How difficult was it to maintain those clusters in the zebrafish vessel system? I'm guessing that cell-to-cell addition is so important that once it's a cluster, it's actually going all the way to being in the system as a cluster and then exiting as a cluster.
So essentially what we did is, so the cells, when we injected them, we modulated whether they were clusters or not by the density of cells that we were injecting. So unfortunately, the heart, so we have to inject right before the heart area of the zebrafish, and the heart is not large enough to fit most clusters. So it's not, what we tried to do initially was to form clusters in vitro and then inject those clusters into the zebrafish. And what we had happen was a bunch of embolism were forming in these zebrafish. They weren't getting through. So to get around that, what we did is we pretty much, to induce the clustering in the system in vivo, we modulated the density of single cells that we were injecting, and then in the circulation, the tumor cells would actually form clusters on their own at a high enough density. So that was what we used to form the tumor. And so usually what would happen is once they formed the tumors in the circulation, then they would remain clusters throughout the process of the extravasation process versus a tumor cell that was just a single cell by itself. So it was tricky, but that was kind of how we got around the issue of not being able to inject clusters themselves into the zebrafish.
I have a cancer biology related question. What happens to those single circulating tumor cells? That's one part of the question. And the second, what if an undiagnosed patient transfuses blood or something like that? What happens to these clusters and single circulating tumor cells? What happens when it's transfused into another person? Is there something that you can say about that? Thank you.
So for your first question, I guess what happens to the single cells? So we see that the single cells, they're still able to travel throughout the circulation and actually exit into new tissue areas. But what we see is that they mostly remain dormant or they don't grow. And what's kind of, like I said before, metastasis is a very inefficient process. So a lot of times, even if the tumor cell can spread and then travel to a new spot, they'll remain in what's known as dormancy. And this happens a lot of times with cancers, is there'll be spread of it, but it'll just be a dormant cancer, so it won't be detectable. And so there are a few different options that can happen. So if a single cell is exiting by itself and it's dormant, it can either remain alive and just dormant, and then at some point, maybe in the future, it can begin to grow or form some type of secondary tumor. It could just die. Sometimes once they'll exit, they just won't be able to survive in that area, and they'll die, or they'll go through some type of apoptosis or anoicus-related cell-mediated death. Or sometimes they'll just remain there and just remain dormant for a long time. Nothing will ever happen, and they'll just remain dormant. But you want them to die. That's the option that you would want. But so to answer your second question, so if someone has circulating tumor cells in their circulation and they give a blood transfusion and another person gets that blood, what's the potential effect? So So usually the good thing about, or kind of the good and bad thing about cancer is that it's an innate, it's part of the body that's acting. It's not like it's a foreign pathogen or body or a foreign article or pathogen that's causing it. It's the body itself or the cells of the body that are causing this. So even if you were to be introduced to someone else's cancer or someone else's cancer were introduced into another person, the body would immediately recognize that it's a foreign tissue or a foreign cell, and it would immediately destroy it. So that's such a good question. And when I first started working with cancer, when I was an undergrad, that was a concern I had because I didn't know about that. And I was working with them, and I think, I was afraid to touch them, or that they would get on me, or that I would accidentally inject myself or something, and I would get a tumor. But then my professor at the time, he kind of explained that and kind of how the whole, with the immune system would be able to immediately recognize them, and they wouldn't be able to survive, or they would die immediately. So that's one good thing, but it's bad for human or for people who have cancer, because that makes it even more difficult to target them because they are cells of your own body.
So given the two models that you suggested for cells exiting the circulatory system, are they both utilized in a normal non-cancer setting by immune cells or other circulating cells?
Excellent question, and the answer is yes to both. So usually I guess one of the methods, the diapedesis method, where they're squeezing through, that's native to mostly immune cells. So white blood cells, when they're in the circulation or when in the system, and there's either a pathogen or some other response, they'll usually they'll undergo the diapedesis method to exit circulation and then reach the pathogen, usually in a kind of a chemotaxic gradient manner, where they'll be able to detect the gradient and they'll travel through the blood vessel to that gradient and then find whatever is causing issues, and they'll attack it or do something. But the other method, the Angiopellosis method, where it's the kind of pushing or the restructuring of the vascular wall and then pushing it out, that was discovered with stem cells. So stem cells that aren't native to the body, that were injected for what's known as stem cell infusion therapy. So there's a type of therapy where you take stem cells, usually adult or maybe some other type of stem cell, for heart disease or even some other diseases, and you can inject them directly into the circulation. And it's been shown that those cells actually have the ability to travel to a damaged area. So say if someone has a heart attack, if you inject cardiac or even other types of stem cells, it's been shown that they're able to home in on that damaged tissue, exit the blood vessel, and then help with the regeneration, or at least mitigation of the disease progression. So that method was first discovered in stem cells to push them out during that process. But that's kind of the normal methods, or the normal cells that have been shown to use both of these methods. It's adult stem cells or adult non-leukocytic stem cells, and then also for the diapedesis method, immune or the white blood cells are known to squeeze out and attack bad pathogens and stuff.
So I was just wondering if the tumor itself or its microenvironment had an impact on the rate at which these clusters or even the single cells can exit the blood vessels.
I think that's a great question, and kind of goes back to what he was asking, is what potentially is different or causing the clusters to be able to perform better. And like I had mentioned there, I think it's really both the clusters having their friends nearby them, and that I do feel like the cluster, it's been shown that cells that are in a cluster versus a cell exhibit different genetic profiles, just like in vitro, just grown in flask or in different settings, not even in an in vivo model. So I think that the cells, cells, they want to be near other cells, and they want to be near cells that are like them. That usually makes them happy, even if you're growing cells in a petri dish or a flask. So if it's just one cell growing by itself versus a group, those group of cells will oftentimes behave much better or grow faster than this single cell by itself. But I think it's really the cells liking other cells around them, and then two, being able to bring almost a piece of home with you for the cells. So the cells bringing, almost if you were traveling, if you were going away from home and you were able to take all the stuff from your room and bring it with you, you might be more comfortable or more enjoyable in wherever you're going if you had a bunch of your stuff and a bunch of your friends with you, versus if you were just alone and isolated and not able to have anything familiar to you. So I think that's probably the biggest or the major component that's going on with the clustering. Is they're just, both the genetic profile is different, but also they're just more happy with other cells that are like them and having increased ability to grow or to proliferate.
I'm just curious that how long after the injection of the tumor cells do you start to see the cells exiting, and what happens to the tumor cells that remains in circulation, and did you notice difference between those that remained and versus those that exited?
So usually with the injection, so what we would do is we would inject the cells, and then pretty much almost immediately after we would inject, we would begin imaging, and then sometimes we would image for up to 16 hours, sometimes even longer. But usually we would see the extravasation or the exiting happen usually within the, I guess, the first few hours, or not usually the first 1 or 2, but like usually after the 2nd or 3rd hour, those kind of time range between the 2nd hour and like the 6th or the 8th, those times were kind of when most or a lot of the extravasation would occur. And with the ones that didn't ever extravasate, we would see that too. And sometimes they would either just get stuck in the place, or sometimes we would see them, they would just be traveling through the circulation. It's very funny, you could just see them like circling around over and over again. Or they wouldn't do anything, or they would just keep circling around. But most of the ones that either stayed in the blood vessel or in the circulation, they seem to be able to, if they were alive at the time when we did the imaging, they seem to be able to at least sometimes maintain their viability, but then after a while, some of them would die as comparison to the ones that would extravasate. So it might just be that they, because like I said, like in the blood vessel, it's like jumping into a monsoon. So it's kind of a harsh environment for cells, especially if you're not a blood cell or used to that environment. But we would see a lot of times, sometimes they just keep circling around and around again, and until we stopped. Yeah.
So these single tumor cells, right? Did you try culturing them?
No, so the only cells that we were culturing were the clusters ones, and I think so. We wanted to, but the difficulty with I guess the single cells, even more than the culture or the cluster ones, is it's very difficult to remove them from the zebrafish and then like culture or plate them and then have them remain viable. So the process, because what we do, I didn't get into it, but when we're getting or isolating the cells from the zebrafish, pretty much what you have to do is you have to disassociate the entire zebrafish and then only get the cells that you're wanting to. And if you have like just one cell from like all those cells, there's a high chance that it's probably either going to die, or you're not going to get the one cell that you were interested in, or it's going to be some other cells that you're getting that are not the single cell. But I think that's the difficult one. But I do think that would be a really interesting thing to do if it could be some way designed to get those single ones that are, I guess, not, or maybe even extravasating, but just the single cells that aren't growing. But I think that's much more even challenging than the clusters, but hopefully maybe in the future.
Once again, I'd like to thank Dr. Tyler Allen for his presentation and all of the attendees for joining us. If you'd like to watch this presentation again or any of the other webinars that are part of Webinar Week, the recordings will be available at genewiz.com/webinar-week.